My name is Vrinda Kalia. I'm a graduate student at the Mailman School of Public Health at Columbia University. My poster today describes a cross-species comparison of metabolic features associated with aggregating tau protein. Tau is a microtubule-associated protein that is found primarily in neuronal axons. Its function is to stimulate microtubule assembly and provide stability, which is important for axonal function and transport. In Alzheimer's disease or other neuropathies, abnormally hyperphosphorylated tau protein leads to the formation of intracellular tangles. These phosphorylated tau, p-tau proteins can be detected and measured in the cerebrospinal fluid. There is evidence of aggregated tau protein affecting metabolism in the brain. Indeed, levels of p-tau in the brain are associated with decreased glucose metabolism. Further, in vitro studies suggest that p-tau is associated with reduced ATP production and imbalanced calcium buffering capacity in neurons. To uncover the global metabolic effect of neuronal phosphorylated tau protein, we use the model organism C. elegans. Specifically, we use the BR5270 strain, which pan-neuronally expresses the aggregating fragment of human tau, and we use its control strain BR5271, which expresses an anti-aggregating form of the protein. To measure the effect of pan-neuronal aggregating tau protein on global metabolism, we use high-resolution metabolomics in C. elegans as developed by our lab in collaboration with the clinical biomarkers lab at Emory University. Very briefly, worms are grown on agar plates where they feed on E. coli as the primary food source. Worms are washed off the plate as they reach the L4 larval stage, which is just before adulthood. They are washed in M9 buffer about 3 times and then sorted into aliquots of about 500 each. Each replicate of the strain is then used for metabolomic processing. Metabolites from each sample are extracted using acetonitrile, and the extraction efficiency is improved by using a bead beater. Essentially, a bead beater disrupts the worm cuticle and allows us to access the worm matrix held within. All samples are injected on 2 columns, the HILIC under positive ESI and the C18 under negative ESI, and again, mass is detected on a thermo-orbitrap instrument. Mass spectral data are extracted using APL-CMS and XMS Analyzer, which are both open source peak-picking algorithms developed at the Emory Clinical Biomarkers Lab. Our preliminary analysis suggests that the pro-aggregating tau strain groups differently from the anti-aggregating tau and the wild-type N2 worms, as seen here in this PCA plot, where PC1 is plotted against PC2 in this pie plot. Further, we ran multiple t-tests for each feature between the 2 groups, and we find in this Manhattan plot where retention time is on the x-axis and the negative log-10 p-value on the y-axis, that about 900 features are significant with an FDR threshold of 0.05. And these sort of significant features enriched multiple pathways that were determined using the Mommy-Chog algorithm hosted on MetaboAnalyst. We used the C. elegans GEG metabolic map as the reference. Strikingly, we see glycerophospholipid metabolism as well as several amino acids that are precursors to neurotransmitters that were enriched. It's interesting since all of these pathways have been implicated in neurodegenerative diseases in the past. Also interestingly, several choline metabolites were matched with level 1 confidence to our in-house reference library. In all 3 of these choline metabolites, we see that there is a reduced expression or a reduced intensity in the pro-aggregating tau strain compared to the anti-aggregating and the wild type. This is interesting because acetylcholine is known to be reduced in patients of Alzheimer's disease, and therapeutics are given to Alzheimer's disease patients that include acetylcholine esterase inhibitors. While all of these results were really interesting, we know that worms only tell us so much about human biology. Therefore, we use data available from patients recruited through the Emory Cognitive Neurology Clinic and the Emory Alzheimer's Disease Research Center. We use these data to determine the association between the plasma metabolomic features as well as p-tau levels measured in the cerebrospinal fluid from each patient. This paper from last year shows the high-resolution metabolomic profile of Alzheimer's disease in this cohort, which here we see that we have about 45 to 50 patients from this cohort that belong to the control group, to a group with mild cognitive impairment, and to a group with Alzheimer's disease. Their mean age is pretty similar, the gender is pretty similar as well. However, in the CSF, the phosphorylated tau levels increase from the control group to the MCI to the Alzheimer's disease group. What we did next is fit a regression model controlling for age, gender, and the batch of analysis. We determined the association between each plasma chemical feature, the log transformed feature, and the levels of p-tau in the CSF that's represented by X1 here. We were therefore interested in the beta-1 coefficient that told us about the association between this feature and the CSF p-tau levels. This Manhattan plot here again shows retention time on the x-axis and the negative log-10 p-value on the y-axis. We see about 391 features that met the p less than 0.05 threshold. And these features were again fed into the mummy jug version 2.0 algorithm using the human reference map. What was interesting is we see lysine metabolism as well as carnitine metabolism, both of which have been associated with neurodegenerative disease in the past. However, they did not reach significance here. What we did next is a little different and still work in progress, is we used the features that were of significance from the human analysis as well as from the worm analysis, and we tried to determine which annotations were overlapping. We found about 10 of them to be overlapping, and we're still following up on them, but a lot of them have been implicated to be associated with neurodegenerative diseases in the past, including cysteine as well as lysine. A lot of work remains to be done. We still want to describe the change in metabolite networks between the two worm strains, as well as compare the metabolic network between human and worm data. With that, I'll take any questions.